ਸਤਿ ਸ਼੍ਰੀ ਅਕਾਲ ਸਾਰਿਆਂ ਨੂੰ। Myself, Dr. Gurpreet Kaur Gill, Assistant Professor Biochemistry, Department of Medical Lab Sciences, Khalsa College of Pharmacy and Technology, Amritsar. Today I am here to present my lecture on diabetes mellitus and regulation of glucose metabolism in the body. As we know that diabetes mellitus is fast gaining status of being an epidemic in the country, India, with an estimated cases of 73 million. WHO has estimated the global prevalence of diabetic cases approximately 14% in 2014. And it has been observed that in India, the urban population, that is 10 to 15% approximately, is diabetic mellitus patients, and rural population is 3 to 8%, that is diabetic. It has been observed that the Indian population is at number 2nd in the world being diabetic. 1 in 6 person in the world diabetic is Indian. So it is important to identify the factors, the factors such as genetic and environmental factors which leads to this disease. So the etiology of this disease, that is type 2 diabetes mellitus, is genetic, which is coupled with certain environmental factors such as obesity, lifestyle, and the stress management, physical activity, which leads to the this emergence of this disease. It has been observed that the poor glycemic control in Indian diabetic population, due to which several complications, whether microvascular complications or macrovascular complications, they takes place. The disease, if it cannot be treated, but it can be controlled, and the morbidity and the mortality rate of this disease is enormous due to the various complications, and it poses a heavy burden on the family members and the society. So what basically this disease is? Diabetes mellitus is of two types. Type 1, that is insulin dependent diabetes mellitus, and type 2, that is non-insulin dependent diabetes mellitus. Diabetes mellitus is not merely a single disease, but a combination of metabolic disorders in which the glucose level in the body remains high for a prolonged period of time, and this condition is called hyperglycemia. The symptoms of this disease include increased hunger, increased thirst, polydipsia, polyuria, that is frequent urination. The euglycemic condition, that is the normal glucose levels in the body, 80 to 140 mg per deciliter, is required for the normal functioning of the body. But in this disease, the glucose metabolism gets upset, gets altered, and leads to two conditions. One is hypoglycemia, the other one is hyperglycemia. If the level of the glucose decreases than the normal, then this condition is called hypoglycemia, in which the cells of the body, they get starved. And in case of hyperglycemia, the level of the glucose gets increased, and this leads to the hyperglycemic state, due to which cellular damages in the body can occur and leads to the complications such as diabetic retinopathy, diabetic nephropathy, diabetic neuropathy, and in some cases there are foot ulcers and the ultimate treatment for the foot ulcers is amputation, which is not an optimal condition for the body, and it makes the body dependent. So it is important to identify the factors, important to understand the etiology and pathophysiology of the disease. But first of all, we have to know about how the glucose is metabolized in the body. So as we know that in our body we have three types of fuels: glucose, fats and proteins. But the body prefers glucose as the primary source of energy. Why? Because glucose upon metabolism gives energy and water through Krebs cycle and aerobic metabolism. Whereas if body uses proteins and fats as energy source, body has to break down first proteins and these proteins and proteins to break down into keto acids, and this leads to a serious condition, that is ketoacidosis. This is one of the microvascular complication, that is the diabetic ketoacidosis, and ultimately it leads to metabolic acidosis. So the glucose is the preferred source for the energy in the body. It has been observed that in non-diabetics, after 5 minutes of meal ingestion, insulin is released, and the effect of insulin can be seen in 2 hours, because the second phase of insulin released is after 20 minutes. So 2 hours postprandial blood glucose can be monitored to assess that how well the body can use the insulin and metabolize the glucose. And the whole chapter of this glucose metabolism is regulated by a very delicate interplay of hormones of the pancreas. The insulin is the peptide hormone which is released by the beta cells of the pancreas, and it lowers the blood glucose levels after the meal ingestion. And the excess glucose, which has not been used by the body, gets stored up by the liver in the form of glycogen. And when the body requires glucose, then liver can release glucose in the body and increase the level of blood glucose and transports in the bloodstream for the normal functioning of the body. Similarly, the third hormone, that is somatostatin, it is also a hormone and is known as the policeman hormone of the body, because it regulates the turning on and turning off of the insulin and glucagon hormones. These both hormones, they act antagonist to each other. These hormones are opposed to each other, and somatostatin regulates the working of these two hormones. Another hormone, that is amylin, it is released in the body in the ratio 1:100 of the insulin, and it is also known as the satiety hormone, and it gives satisfaction to the body, and it delays the gastric emptying, which gives satisfaction to the body for not eating too much. So it helps in the brain meal connection somewhere, and it reduces the immediate spike of the blood glucose in the body. There is another biochemical, that is incretins. Incretins are peptides which are released by the intestine, and this is also helps in the sudden increase in the blood glucose spikes. So it has been seen that our body is designed in such a way that there is an interplay of hormones and biochemicals which so beautifully regulates the level of glucose in the body, which is ultimately requirement of the body for maintaining and performing the functions in the body. So we can say that liver is such an amazing organ which knows how much glucose to store and keep, how much glucose to break down and release. And it has been noted that approximately 10% of the glycogen can be stored by a healthy liver, and muscles also, they can store up to 1% of the glycogen molecules for the immediate needs of the body. Hyperglycemic condition is a diseased state if it persists for a long period of time, and this can poses several threats to the body by emergence of the complications or the secondary microvascular and macrovascular complications in the body. So decreased insulin production and increased insulin resistance both are harmful for the body. So the hyperglycemic condition causes certain cellular damages in the body in case of diabetes mellitus diseased state, and how this condition leads to the damage. First one is glycation. Excess glucose in the body leads to the glycation of the proteins present in the body. For example, hemoglobin. Hemoglobin gets glycated by the glucose if it is present excess in the body to produce HbA1c, which is a hallmark molecular diagnostic indicator for the onset of diabetes mellitus in the body. So the glycation products, when accumulated in the body, they disrupts the endothelial function of the body and leads to the complications in this disease. And the second route of damage is through the activation of protein kinase C. Excess intracellular level of glucose activates the protein kinase C, which releases certain inflammatory markers and releases cytokines, homocysteine in the body. Third is there are certain intracellular activities in the cell which protects our cells from the free radicals and oxidative damage. But if the level of the glucose in the cell or in the body is higher, then it will reduce the effects of that intracellular activities, which leads to the oxidative damage to the neurons. And this will pose serious complications in the body. And these complications need to be reduced by the appropriate suggestive preventive measures for the management of the disease. So the clinicians may be targeted to implement certain pre-screening and management practices so that this epidemic can be managed in the country. Okay, so the first one should be the early screening and detection of pre-diabetes. Secondly, the level of insulin should be maintained in conjunction with the hypoglycemic agents, early initiation of the insulin so that the glycemic control can be done in the patients for the prevention of the further complications. Third and the most important is the lifestyle management. The preventive measures should be taken and the lifestyle should be managed, modified for the prevention of this epidemic in the country. So this was all about diabetes mellitus and regulation of glucose metabolism, and this lecture would be helpful for the students of BSc Medical Lab Sciences MSc Medical Lab Sciences. Thank you very much.